Thank you very much, Anthony. Invite the speakers and Michael Chapman to join the stage. So I think we're well drilled now. We have a team of dynamic bearers of microphones, so do raise your hands to speak, and please identify yourself with your name and your affiliation. We have questions online and a few to start us out, but please do raise your hands. Maybe I'll just start with the first question while you raise your hands and put our colleagues in place. So Michael, welcome. So you're from, as I know very well, we've obviously worked together for a long time, from NHS Digital, where for the last few years you've been the Director of Research and Clinical Trials. Some people will know NHS Digital very well in the audience. Others may know nothing about it. Would you like to say maybe a few words about how it fits into this landscape and how it offers something important in this space, but maybe also a self-critical reflection on what it might be able to do better?
Absolutely. Thanks, John, and thanks everybody for having me here. So, as it says on the slide, I am Mike Chapman. I'm formerly of HDR UK, now of NHS Digital. And NHS Digital is, for the next 8 weeks or so, and I'll say why that is, the central data and technology body for the NHS in England. So a major part of the organisation's role is to be the central safe haven for patient data, curating, collecting, linking, and then disseminating to authorised users health data for research like that by UK Biobank. And it's an organisation that can do some amazing things. You know, my job there is to try to ensure that data is used for research. And at any one time, as well as UK Biobank, we have about 500 data sharing agreements with different research studies. So there's a vast amount of research that is founded on the data that's being collected every day from NHS patients and then used to improve health and care. To give some examples, as well as giving out data to people, we are increasingly building services on top of that data. What I've been trying to do for the last couple of years at that organisation is try to ensure we can make better use of the data. So we've got services now for clinical trialists to help assess feasibility of trials, to help recruit trials, and to help follow up people who are part of trials or consented cohorts. And that's an incredibly powerful use of national data. We've finished recruiting now to a study called NHS Gallery, a study of multi-cancer early detection technology. We recruited 140,000 people with the investigators in 9 months. That's the fastest ever large-scale recruitment to a randomised trial in the UK. And that's what you can do with national data. With HDR UK and with the British Heart Foundation Data Science Centre, we hurriedly set up during COVID a secure data environment, a trusted research environment for people to be able to access population-scale data without needing consent and in a very secure fashion. We've now been building that out. It's got about 200 users in it, working on 56 different studies as of today. And again, that's enabling access to data in a way that's just not possible if you have to hand out copies of population level data to people. So it's an organisation that can do amazing things. There are definitely places where it could be better. We've heard about one of them earlier, the perennial frustration of how do we enable studies like UK Biobank to access data from primary care? And it's a really sticky issue and one that we haven't cracked yet. We're still working on it, but it's hard and it's going to take us some time. Final point, and then I'll hand back, John, so I don't want to monopolise this, but I said for the next 8 weeks or so, part of the ongoing structural changes to the NHS Digital will be merging with NHS England to give one central body for the NHS, well, fewer central bodies for the NHS in England. One of the explicit intentions of that merger is to improve use of data by the healthcare system for 4 purposes: direct care, population health, planning, and for research and clinical trials. So there is an ambition to make this better. I'll stop there.
Well, thank you. Before we go to the floor, just to follow up on the primary care records, we did get a question online about that, and maybe it's a question that's somewhere between you and Naomi. The question is, when will primary care records be linked to the remainder of the UK Biobank participants in a way that can be used on an ongoing basis?
So, I mean, we may need Naomi to answer on this one. The when, I can't give a timescale for that, or coming from NHS Digital. And the reason I can't give a timescale for that is that we need to land the merger with NHS England. We need to demonstrate new structures we'll have in place to protect patient data. and we then need to go back out to patients and to the general practice profession and show that we can use that appropriately. So we've got work to do before we can actually set a timescale for that. But I don't know if the work you're doing directly might go faster now.
Well, that's the million-dollar question, isn't it? When will these data be available? So we're hopeful that we can find a shorter-term solution whilst waiting for the longer-term NHS digital one by having a direct mechanism, a bit like the emergency legislation that was introduced to enable COVID research, to enable the flow of GP data automatically from the system suppliers to UK Biobank for these 500,000 consented participants. So it's something we're still in discussions with, but we have reasons to be optimistic that we can find a shorter-term solution. As to the exact timelines, nobody really knows, but we're hopeful.
Thank you. Microphone number 4, please.
Hi, my name is Wahab Al-Shalal. I'm a research assistant at the MRC Epidemiology Unit. My question is prompted by the All of Us cohort, and it was related to the Fitbit data. And I wanted to ask, what is the potential for using smartphone derived data from participants to try to fill the gaps between from non-wear during Fitbit data? And it could be applicable to the UK Biobank and accelerometer data. Is there potential for that, or is it as tricky as getting the primary healthcare data?
It's a great question, and I'm really glad that you asked about the idea of another type of measure, because I think it's very powerful. In our case, what I'm showing you were data that our participants linked through the app or website to their Fitbit accounts, and it's sharing some of that primary data from Fitbit. People also share in other kinds of information, so through Apple Health Kit, and we also get step information from that. That can also come through other devices. They are not within the All of Us. It's not released as a research resource yet. We haven't done any curation on that kind of data, but there are other ways in which people do provide those kinds of data. And so I think it is a really powerful direction going forward of amalgamating a number of different resources into those common data sets. Now, I talked a lot about data harmonization on electronic health records. I think there's a similar problem of data harmonization on digital health technologies. Probably steps or something everything provides, but once you start to get beyond that, I think there's quite a bit of variability.
Thank you.
Number 1.
Yes, thank you all. Kezia, about the image-based molecular classification, you are trying to find some correlation between images and genetic markers, if I am true. And what kind of genetic data are you taking into account? I mean, what do you think? What's the major genetic variation that affects histopathological type of the tumor?
Could you just briefly identify yourself?
Oh, sorry. I'm Shahryar, bioinformatician from UCL.
Thanks. So the data I was showing that was sort of directly replicating molecular changes in tumour from the ESCORT consortium, that was based on a molecular classification of colorectal cancer, where I think it was a combination of sequencing data and a sort of variety of molecular tests that they were specifically trying to generate an image-based classifier that could replicate that classification. So that was a sort of very specific thing they were trying to replicate. In terms of sort of wider potential uses in the future, I think histopathology is in many ways an undertapped resource. So obviously I would say this because I'm a pathologist, so I recognise my bias here. But I think you could argue that the morphology, what the tumour looks like under the microscope, is really an end phenotype. So it's a combination of all the germline genetic mutations, the somatic genetic mutations, the epigenome, the tumour stroma microenvironment, all the things that we might be interested in. And I think there's already been a lot of digital histopathology work of people trying to come up with image-based algorithms or machine learning to pick up some of that. But I think there's still a huge amount of potential in the future.
Thank you. Number 3, please.
I'm Dawood from the UK Biobank imaging team. Question for Josh, please. Do you have any plans to add imaging to the All of Us study? And if so, could you tell us a little bit about what type of data you would collect? Thank you.
I'd love to hear more opinions on what we should add, actually. So we are a growing resource, and I think we are still working out many plans for other kinds of data that we will collect beyond what we've already done. And certainly imaging is one of those other things that we have talked about. We are having discussions internally within the NIH now. Probably the first it's a proxy for imaging data, but the first kind of imaging data we get will probably be the radiology reports, the text of the radiologists read on the images. That'll probably be the first thing that makes it in as we work through capturing that information from electronic health records and then de-identifying it. But it's also possible that there'll be some ancillary study that generates some other kind of imaging data beforehand. So we're looking at many of the things that have been discussed today for sure.
Thank you. Number 2.
Hi. Andrew Morris from HDR UK. So first of all, 4 terrific talks, and great to see Michael Chapman too, who was heroic during the pandemic. So my question is about knowledge management and open science, because we're going to see a proliferation of algorithms, phenotypes, analytical pipelines. How do you feel we're approaching knowledge management internationally, and are there opportunities to do better?
Well, I'll start. Well, I think the first step is being able to openly share your results and the underlying methodology used to generate those results so that others can replicate your work or build on the work that you've done. And I think in UK Biobank, we've made a good start on that by researchers are obliged to return those results back. I think building on from what Josh said about having common coding standards in OMOP or other coding classifications across the UK and the US and other population-based cohorts will help massively in researchers being able to look at phenotypes and say, yes, I know what that is using a common standard. But there's so much more we could do. We're so used to working in silos, particularly within our own countries, EMR systems. But I think we are making some good steps in terms of talking with our international colleagues. The scientific world is a small space. And I think we are making strides, but certainly there's much more that we can do in terms of communication, open source, publishing our methodologies and our algorithms for how we define phenotypes is exactly what we should be doing.
And perhaps if I can add to that, I agree entirely. I think this starts with working in the open, and as an organisation collecting and curating these data sets, part of what we're starting to do is publish our statistical methodology particularly, but also our code much more openly so people can see. And in fact, Andrew, I was talking to one of your team earlier today about how we get that end-to-end visibility of how is data being collected, how is it being transformed through that process. And so people can then choose whether they accept those transformations or if they want to apply something differently. The second thing that we can do is encourage and enable people to work collaboratively and make it easy to work collaboratively and harder to not work collaboratively. And we can do that with a carrot approach. We can design things. We're trying to design the secure data environments we're setting up to enable code sharing and collaborative working and enable that rather than prevent it. We can also apply a little bit of a stick if needed, because at some point people are going to want to take results out of those environments. And one of the questions we can ask them is, okay, have you shared the code underlying these results? And if you haven't, what's the justification why you wouldn't do that? So we can start to exercise some control. We're not there yet, but it's something we can do if needed.
I'll just add quickly, I think there are a lot of areas we've made a lot of progress. I'll give an example. When first writing EHR algorithms to find conditions, the first time I tried this, I wrote pseudocode and thought that would be helpful. And it wasn't at all, this is 2007, it wasn't at all helpful. It ultimately took people about 2.5 years to execute off of like flow charts I created and lists of terms, and they were able to replicate those kinds of findings. You know, and I gave, you know, fast forward to my example with my PhD student, you know, he took that OMOP code and then, you know, in a week had results from, you know, all of us, which included application time to the, you know, resource. So, you know, there are some examples of making progress on, you know, sort of phenotype algorithms. And, you know, to Naomi's point, one of the reasons we picked OMOP is there's actually multiple different, you know, standards on data models, but they're not, they tend to be within a country, or they tend to be more limited. And so I was really an advocate to going to, you know, the broader data model, which encompasses the EHR and could be more useful. And I'll just give you kudos to representing PRSs, for instance, in a way, and putting that as a resource that you could find. I think it's really hard to assimilate the data that comes in from research, and then make that re-available. I think it's something is underexplored now.
So on that same theme, Josh, so UK Biobank was praised in an editorial in Nature a couple of years ago for providing a gift to the world with its open access data model, and has made big efforts, recent initiative in Africa to try to widen it further to users. I know that efforts are being made in all of us. It has a different governance model and a different set of restrictions. Would you like to comment on the plans for expanded access?
Yeah, I'd be happy to. I mean, I think the UK Biobank really has been a transformational leader in this space. You know, all of us always begun with the idea that we would expand and continually try to facilitate research being done on the platform, including international access. And so what we wanted to do is we wanted to get data into the hands of researchers as quick as possible, while also building trust with our researchers. So as we continued to build the resource, you know, we launched our research platform in 2020 during COVID, while we were also doing a whole bunch of stuff according to COVID. And at the time, you know, it was it was limited really towards US nonprofits. And we really wanted to engage. I mentioned we have a really underrepresented, we've really focused on recruiting underrepresented population in the US. And that became a really hard time for trust in the United States, as you know, COVID hit them particularly hard. There's a lot of things that happened in our country at that time, and so we have carefully built up, and we have already expanded access to the program in terms of right now still U.S., but the populations that can access it within the U.S. have expanded from just mainly academic to academic, educational, for-profit, and nonprofit in those domains, and healthcare domains. And so one of the next steps would be as we expand to international audience. So that careful project or process of creating the release, building trust with our participants as things are done, as science is done, that's respectful and non-stigmatizing. And, you know, it's something that we're working on, and we're looking forward to expanding access to.
Thank you. So I realize that Anthony's a bit underutilized at that end. So if I may ask a question. So Paul Matthews seemed to be suggesting that the eye was the front of the brain. I think you were arguing that the brain was the back of the eye. Could you comment on what you think is the potential for technologies like the optical coherence tomography, which at the moment the retinal nerve fiber thinning, I think, gives us non-specific information about not only Alzheimer's disease, but it also thins in the presence of other neurodegenerative diseases. Do you see the possibility of a more one-to-one relationship with some kind of imaging biomarkers and particular brain diseases emerging?
So it's an area that people are very excited about, so I certainly don't want to dampen any excitement. To date, the associations between imaging biomarkers and neurodegenerative diseases, for example, I think have been relatively weak, but they're definitely there. They're reproducible. We're seeing it in multiple cohorts. And so I think building on that, I think we need to further refine how we're going to best get more information out of the images, probably using machine learning methods to be able to better classify and better predict. I don't think we're there. I don't think we're there yet. So I think the 60,000 extra participants that are going to be imaged is going to be hugely beneficial. I think in later rounds of eye linkages, if the first round is successful, we'll be able to bring in more OCT scans that way. So it's an exciting area.
Great, thanks. We've got 5 more minutes, so maybe a few more short, sharp questions and brief answers. Microphone number 3, please.
Hello, my name is Andrew Nigoro. I'm a data scientist at Relations Therapeutics. As a matter of fact, I also have a question for Anthony. When I looked at this visualization where you could follow kind of the vision impairment over time for different patients, I was under the impression that for some actually the vision improved for over maybe 10 or more years. So I thought that was quite curious. And this was kind of related to the fact that I think we spoke a lot during the day about risk scores, but I was wondering how amenable or important is, in your opinion, doing also research in terms of protective factors with UK Biobank data or in general, really.
So the improvement's interesting, and it's sometimes just learning to do the test better. It's actually quite a challenging test to concentrate for 5 to 7 minutes on each eye, pressing a button every time you see a light, and sometimes people improve. So that's there's an element of that. There are also other things that can improve the visual field. For example, having cataract surgery. In terms of should we be looking at protective factors as well as risk factors, that's something actually that Mendelian geneticists often ask me, and I think when you're talking about common diseases with common variants, each risk factor both protects and increases the risk. They're both, so it just depends which way round you look at it. So, of course, we want to be looking at being preventive as well as just and getting clues as to how we can prevent disease, as well as just predicting who's at highest risk, so we can focus our resources on them.
Thank you.
And we have 2 questions up front that might be our final 2 questions. One here and one here, please.
Hi. Thanks, Anthony. Another question for you here. You think, so with regards to linkage, right? So you mentioned that the ophthalmology seems to be a great example. Everything seems to be harmonized, standardized, etc. What were the challenges? So I'd like to hear, what can we have learned from your experience that we can actually take to other linkage exercises, right? Consent was a problem? I don't know.
So the linkage hasn't started yet, and we're hoping to carry this out next year. Sorry, I wasn't clear about that. What I foresee is the biggest challenge actually is going to be is getting approvals from all of the data controllers. So for example, the National Diabetic Eye Screening Programme, actually the 2 software providers are actually the data controllers and Caldecott guardians for many of the screening programs. So that's a bit easier, but there are still other ones where we'll have to get individual approval. Same for the Medisoft centres. Medisoft are going to help us run the linkage, but we're still going to have to get approval from each of those 15 centres we're going to. What I can say is to date, we've done larger projects than that. We've got Caldecott Guardian approval for 27 centres, for example. And so far we've got a really good experience and track record. I'm sure there's going to be a lot of learning, and we'd be very happy to share that as we're going along, and also welcome any advice or any good ideas from the audience.
I mean, just more generically on that, with all the linkages we've done, I mean, our experience so far, I mean, consent isn't really an issue because we do have explicit consent up front from all 500,000 participants when they were recruited. We have consent to link to their health-related records. So that isn't the issue. A lot of the time goes into making sure the regulatory approvals are in place, the bureaucracy is all done. That takes an awful lot of time. And then once you do get your hands on the data, there's an enormous amount of work that goes into understanding what data has been sent, are all the data fields documented, doing all the data checks. Do we understand what all of the coded fields mean? It's no good us releasing data if we don't know, if we don't have the appropriate data dictionary. And so all of that data curation work it can be very time consuming. So I would say those are the generic challenges we face.
So, and I think that the latter ones are the ones that I think I'm more confident on, because we already have the data dictionary and they're used to those extracts. So fingers crossed.
Indeed. Final question, microphone 4, please.
Thank you. Hi, I'm Michael from Sydney University. I also run an analytics and software company. Thanks a lot. It was really, really interesting. I have a question around the sort of intermediate layer between the raw data and kind of papers, research results. Is there a vision or an appetite, or does it already exist in terms of kind of data visualization, reporting kind of software that would enable a wider group of people to access some of the power of the UK Biobank? Is that on the horizon, or any thoughts on that?
Well, we'd love to hear from people who are interested in data visualization, for absolute sure. I mean, it's an area which is just starting to regain real traction now. I mean, you know, our job here, we build the resource, we make the data available, but there's a whole other aspect which is about understanding the data, and data visualization is a key part of that. So, I mean, we're very open to suggestions, and on the research analysis platform, there are so many tools that can be incorporated into that platform that I think it's really ripe for these kind of data visualization tools. So we're very, very open to have conversations about that, for sure.
Thanks, Nice. So on that note, we'll draw this session to a close. It's been a cracking one. Join me in thanking the panelists and the speakers. Thank you very much.